All right, so the way that we know how old all of this fossil evidence is, for example, when we're talking about the stromatolites, the first bacteria fossils, is through radiometric dating. And the basic way that this works is that um, all elements have different isotopes, which means that there's a different amount of neutrons in their nucleus, which makes some of the isotopes heavier or lighter compared to other ones. What's happening over time is that the ratio of heavy to light isotopes is constantly changing as the isotopes decay. And when this radioactive decay occurs, we can then use that to time the age of a fossil. And so it's just sort of an aside is saying, well, how do we know all these rocks are this age? How do we know there wasn't some, catastrophic flood that, put all of the fossils up on the top of mountains? Well, that's why. We can calculate the number of isotopes, and if we know how much they're decaying, we can go back in time and figure out how old those fossils are. Remember that fossils are typically laid down in sedimentary rock. Um, and that the newer fossils are on the top and older fossils are on the bottom, which means that life forms that look like us are found higher up in the sedimentary rock, and more different looking things are found further down. The text talks a lot about the geological record, and I invite you to go take a look at that. The different ages, some of the main points in different parts of the Earth's history. There are a couple of extinctions that I want to focus on, mainly because one of them you've heard of before, and one of them you probably haven't heard of before. So the Permian extinctions are often known as the mother of all extinctions, and the reason why they're so important is because basically a number of different factors we think came together to cause this extinction. And when it happened, a huge number of families were lost. And so, what we think happened in the Permian extinctions was that Pangea comes together. So, I know that in geography class, you guys have learned about continental drift and Pangea. So, as that's happening, Pangea is coming together. All of the shallow seas that are in between each of the continents start to dry up, and obviously all of the oceanic life that is existing in those shallow seas no longer exists after that point. So that kills off a lot of the marine life. At the same time that Pangea is coming together, there's also obviously going to be a change in wind and air and water currents. And what that means is that the climate is going to change drastically, and especially the interior of Pangea is going to become very dry. And so this climate change brings about another layer of extinctions. At the same time, we also think that in Siberia there was a huge amount of volcanic activity, and so that those volcanoes were spewing ash into the air, which would have had a cooling effect on the Earth, but they would have also, over the longer term, been spewing CO2 and other greenhouse gases into the atmosphere, which would have then caused a subsequent heating of the Earth. So there would have been a cooling followed by a heating, and that would have caused an extinction of a large number of animals. And then you also have the heat causing the glaciers and the ice caps to melt. And when that happens, you have flooding in lower lying areas. And so again, you have another level of extinction on top of that. So the Permian extinctions are a pretty important event that I find often students coming into the AP class haven't heard of before, but it was far more serious in terms of the total number of families that went out of existence than the Cretaceous extinction, which many of you are more familiar with. So if you're just looking down here on the graph, comparing the number of extinction or the number of families that go extinct between the Permian and the Cretaceous extinction, quite a big difference. The thing that you can also notice, though, is that every time there's an extinction event on these graphs, the overall diversity of life on Earth goes up. So Each extinction event is followed by adaptive radiation, which we covered in the unit on evolution. So the Cretaceous event, as many of you are probably aware, we believe was caused by a comet that hit the Yucatan. Although there is still a lot of debate about what actually caused the extinction of the dinosaurs, and there is a lot of evidence to suggest that something was already causing the dinosaurs to suffer before that comet ever hit, and it was kind of just like the final nail in the coffin. Although in the last couple of years there have been a number of paleontologists who have started to say that that's not correct either, that many groups of dinosaurs were still thriving at the time that the comet hit. So there's a lot of debate about the Cretaceous extinction and whether it was even caused by a comet or not, and then after that, were the dinosaurs already having problems, or were they fine? Alright, so in the fossil record we can find a lot of early evidence of life. So we've talked about the stromatolites, those extensions in the tidal pools. We also know that at some point in Earth's history, about 2.7 billion years ago, we start to have the first concentrations of oxygen form, and that caused the precipitation of iron out of the water. And so you can see here in the sediment down in this image the banded iron patterns, which is showing us that oxygen has started to be released and it's starting to pair up with iron in the water. Once all the iron's out of the water, then it starts to be released into the atmosphere, causing probably the first mass extinction of animals unable to deal with an aerobic environment. We also know that all life shares electron transport chains, which means that the electron transport chain was something that was found in the organisms before we had the last common ancestor. So we're going to talk about the electron transport chain quite frequently here in the next couple of chapters, and so it's just an important idea to keep in our minds. It's used in both cellular respiration and photosynthesis. And I think in the next slide what we're going to look at is how did eukaryotes form through the endosymbiosis of both mitochondria and chloroplasts. So the endosymbiotic theory says that mitochondria and chloroplasts inside of eukaryotic cells actually used to be solitary prokaryotes that somehow made their way into eukaryotic cells. We're not sure whether they would have been prey or whether they would have been parasites, but once you have the mitochondria and the chloroplast entering into these cells, you're going to have a fundamental transformation in the way that eukaryotes are able to carry out their business. So looking down at this diagram, what we think happened is you have an ancestral prokaryote which increases in size. And remember that when you have an increase in size, you have a problem with your surface area to volume ratio. And that means that it's going to be harder for gases and other nutrients and waste to either come or go. And if that's the case, then what you need to have is sort of infoldings to maximize the surface area to volume ratio. And so these infoldings start to form and will eventually become the endomembrane system, which you'll remember from cells is the endoplasmic reticulum, the Golgi bodies, the nuclear envelope, and all that sort of good stuff. So at some point, there is a prokaryotic cell that resembles a mitochondria that makes its way into our ancestral eukaryote. And once you have a mitochondria inside of, well, an ancestral mitochondria inside of an ancestral eukaryote, what you have is the ability to take a sugar like glucose and break it down very efficiently. And in breaking it down very efficiently, you're able to generate a huge amount of ATP versus not using a mitochondria. So when we look at cellular respiration, we'll see that the difference between breaking down a molecule of glucose without a mitochondria anaerobically inside of a cell only yields about 2 ATP, whereas if you break a molecule of glucose down using a mitochondria, you yield 32 to 36 ATP. So that's a huge difference in efficiency, and really the mitochondria, once it enters, allows the cell to be very efficient, and by being efficient, it lets it be big, and it lets it do bigger things than what a prokaryotic cell could do. At some point after the introduction of this ancestral mitochondria, some of the cells had chloroplasts in a similar way enter into the cell. We don't know again whether this was because they were a prey or because they were a parasite, but somehow they enter, forming a symbiotic relationship with this cell. And over time, what we have is basically the chloroplast becoming a what do you call it? A functional organelle inside of the cell. Again, the advantages are huge for eukaryotic cells that have this photosynthetic prokaryote inside, because now they're able to use the energy of the sun to produce glucose. And once you can do that, you have a supply of basically an unlimited supply of energy. And now on top of that, because they also had the mitochondria, we know that every glucose molecule of glucose being produced by the chloroplast is going to be used very efficiently by the mitochondria. We know that chloroplast came in second because, remember, all eukaryotic life, well, not all, but generally speaking, almost all of eukaryotic life has mitochondria. When we look at the protists, you'll see that certain protists have different types of mitochondria, and so there's some variations there, but generally speaking, we can say that they all have mitochondria inside of them, and we know that not all eukaryotic life has chloroplasts. So we therefore then know that chloroplasts were coming in second in this process of endosymbiosis. Remember that, well, yeah, we'll save that for the next time. So what's the evidence of the endosymbiotic theory? How do we know that the mitochondria and the chloroplasts at one time were independent organisms, independent prokaryotes? Well, we know that they're about the same size as prokaryote. Their membranes are similar to those of prokaryotic cells. When they are being duplicated in, let's say, a G2 phase of mitosis, where the cell is preparing for division, all the mitochondria and chloroplasts, they must also duplicate so that each subsequent cell has the correct number of these organelles. And when they split similar to binary fission that's happening in bacteria. So the way that they divide is very similar to prokaryotes. They also have their own circular DNA. So you may have heard before that mitochondrial DNA. So the mitochondria has its own DNA. The chloroplast also has its own DNA, and you'll remember that prokaryotes have circular DNA, which we find in both of these organelles. They also have their own ribosomes. Well, heck, I mean, if they have their own DNA and they're making their own proteins, you need to have their own ribosomes, and that these ribosomes resemble prokaryotic ribosomes. Remember that this has to do with the two subunits that make our snowman-like looking structure. At some point, we have eukaryotes coming together and forming multicellular life. And we think that the first multicellular life was actually sort of colonial eukaryotes. And once you have multicellular life, it's not like all of a sudden that's hugely successful. And we think that might be because at one point there's evidence of something called the snowball Earth, where we find glacial sediments up until the land masses that would have been at the equator. So if the Earth, even if there was multicellular life, but the Earth is covered in an ice cap, almost the entire surface of the Earth, then that would mean that there wouldn't have been a lot of photosynthetic activity that couldn't have really supported large multicellular life forms. But once we get to the Cambrian explosion, there's a huge explosion in the diversity of life, and we're going to come back to the Cambrian explosion later. But basically, most of the different types of animals that we're used to seeing, their origins go back to this Cambrian explosion. And at some point, about 500 million years ago, we have the first plants coming onto land. All right, I'm going to just stop here, and then we'll come back to this in a little short portion of part 4 of chapter 26.